Hello viewers, welcome to my channel. In continuation with the previous episode, today we'll go through the history and development of microbiology. Stay tuned in this video. So the first question is, the term mutation was coined by Hugo de Vries, Darwin, Lamarck, Pasteur. And the correct option is Hugo de Vries. Now let's discuss about the mutation. Mutation is the change in the genetic material of an organism or virus. DNA and RNA constitute the genetic material in all living organism or virus. The next question is, compound microscope was discovered by option 1 is Antony von Leeuwenhoek, option 2 Pasteur, option 3 Jensen and Hans, and the fourth option is none of these. And the correct option is Jensen and Hans. Hanging drop method for motility study was first introduced by Robert Koch, Louis Pasteur, Jenner, Leeuwenhoek. And the correct option is Robert Koch. Robert Koch introduced this method in 1878. In hanging drop method, a very small drop of bacterial suspension is hung from the center of a covered slip into the cavity of the cover slide. The hanging drop is observed under the microscope using oil immersion objective. The motile bacterial cells can be seen to have erratic movement in the surrounding medium. The next question is, Pasteur first conducted fermentation experiment in option 1 is milk, option 2 beet juice, option 3 food material, and option 4 is none of these. And the correct answer is beet juice. Next, who provide the evidence that bacteriophage nucleic acid but not protein enters the host cell during infection? Option 1 is Hershey and Chase, option 2 Hershey and Totem, option 3 Liderberg and Totem, option 4 Hershey and Macleod. And the correct option is Hershey and Chase. The next question is, hybridomer technique was first discovered by Kohler and Milstein, Robert Koch, D. Herrele, Landsteiner. And the correct option is Kohler and Milstein. Hybridomer technology is one of the most common methods used to produce monoclonal antibodies. In this process, antibody producing B lymphocytes are isolated from the mice that are immunized with specific antigen, and it is then fused with immortal myeloma cell lines to form a hybrid cells called hybridomer cell lines. Griffith in 1928 reported the phenomenon of transformation first in Haemophilus influenza, Bacillus species, Pneumococci, E. coli. And the correct option is Pneumococci. Griffith, in this experiment, Griffith used both virulent strain and avirulent strain of Streptococcus pneumonia. Next question is, serum sickness reaction was discovered by M. Arthas, von Perkett and Bela-Schick, Rickett and Porter. And the correct option is von Perkett and Bela-Schick. The next question is, the term bacteriophage was coined by D. Herrele, Todt, Pasteur, Robert Koch. And the correct option is D. Herrele. Although the credit for discovery of bacteriophage is given to both Todt and D. Herrele, the term was coined by D. Herrele. So what is bacteriophage? Bacteriophage is a type of virus that infects the bacteria. In fact, the word bacteriophage means the bacteria eater, because they destroy their host cell. All bacteriophages are composed of nucleic acid surrounded by a protein structure. The next question is, viral infection of bacteria was discovered by option 1 D. Herrele, option 2 F. W. Todd, option 3 Bejering, and option 2 Janusk. And the correct option is Todd. The next question is, Treponema pallidum was discovered by option 1 Sordin and Hoffman, option 2 Louis Pasteur, option 3 Barge, and option 4 none of these. And the correct option is Sordin and Hoffman. Basically, Treponema pallidum is a bacterium. It's one kind of spirochete. It is the causative organism of syphilis. Syphilis is one kind of sexually transmitted disease. The next question is, diphtheria bacillus was discovered and identified by option 1 Claves and Loeffler, option 2 Sordin and Hoffman, option 3 Todd and D. Herrele, and option 4 is Krebs and Lippmann. And the correct answer is Claves and Loeffler. Diphtheria bacillus is also known as Claves and Loeffler bacillus according to the discoverer. Transduction discovered by Gindar and Littleberg, Todd and D. Herrele, Hershey and Chase, and the fourth option is Totem. And the correct option is Gindar and Littleberg. Gindar and Littleberg discovered transduction in 1952 in Salmonella. Now, what is transduction? Transduction is the process by which virus transfer its genetic material from one bacterium to another. Conjugation was discovered by Littleberg and Totem, Todd and D. Herrele, Hershey and Chase, Claves and Loeffler. And the correct answer is Littleberg and Totem. They discovered that F factor, that is the fertility factor, can move between E. coli cells and proposed the concept of conjugation. Mycobacterium leprae was discovered by option 1 Robert Koch, option 2 Hansen, option 3 Edward Jenner, and option 4 is Louis Pasteur. And the correct option is Hansen. Mycobacterium leprae cause leprosy. Leprosy is also known as Hansen disease because it was discovered by Hansen. Who discovered prion? Option 1 is Stanley Prushiner, option 2 Carleton Gujdasque, option 3 Totem, and option 4 is Theo Danier. And the correct option is Stanley Prushiner. Prions are misfolded protein with the ability to transmit their misfolded shape onto normal variants of the same protein. They characterize several fetal and neurodegenerative disease in humans and many other animals. The next question is, who discovered viroid? Option 1 is Stanley, option 2 Theo Danier, option 3 Stanley Prushiner, and option 4 Rouse and Yersin. And the correct option is Theo Danier. Now, what is viroid? Viroids are basically a small single-stranded circular RNA that are infectious pathogen. Unlike viruses, they have no protein coating. The first recognized viroid was the pathogenic agent of the potato spindle tuber disease. It was discovered, named, and initially molecularly characterized by the plant pathologist Theo Danier. Who is the founder of virology? Option 1 Ivanovsky. Option 2 Stanley. Option 2 Todd. Option 4 is none of these. And the correct option is none of these. Ivanovsky. Who discovered streptomycin? Option 1 is Waxman. Option 2 Fleming. Option 3 Avery. And option 4 is Kerry Mullish. And the correct option is Waxman. Streptomycin is an antibiotic used to treat a number of bacterial infections. The mode of action is it binds to the 30th ribosomal subunit, thus inhibit the protein synthesis. The next question is, who demonstrated that open tubes of broth remained free of bacteria when air was free of dust? Option 1 is Spallanzani. Option 2 John Tyndall. Option 3 Francisco Ricci. And option 4 is Pasteur. And the correct option is Spallanzani. The next question is, who discovered diphtheria toxin? Option 1 is Roux and Yersin. Option 2 Anthony von Weyering. Option 3 Loeffler. And option 4 is Oxton. And the correct option is Roux and Yersin. Weyering also discovered diphtheria antitoxin. Here the question is diphtheria toxin. So diphtheria toxin was discovered by Roux and Yersin. Loeffler, who discovered diphtheria bacillus. Who discovered genetic code? Option 1 Nierenberg and Kodana. Option 2 Watson and Crick. Option 3 Hershey and Chase. Option 4 Metchnikoff. And the correct answer is Nierenberg and Kodana. So what is genetic code? Genetic code is a sequence of nucleotides in DNA and RNA that determines the amino acid sequence of protein. Who first introduced the acid-fast staining? Option 1 is Paul Ehrlich. Option 2 Jill Nielsen. Option 3 Christian Gram. And the correct option is Paul Ehrlich. Some brief notes about acid-fast staining are here. So first, Jill Nielsen staining is a type of acid-fast staining that was first introduced by Paul Ehrlich. It is a bacteriological stain that is used to identify the acid-fast bacteria. Now, what is acid-fast bacteria? It is a group of bacteria that show acid fastness. So, acid fastness is the ability to resist decolorization by acid during staining procedure. Acid fastness of the mycobacteria is due to high mycolic acid content of their cell wall. The next question is, who discovered monoclonal antibodies? Option 1 is Rodney Porter, option 2 Jacob, option 3 Kerry Mullish, and option 4 is Kohler and Milstein. And the correct option is Kohler and Milstein. A monoclonal antibody is an antibody made by cloning technique of white blood cells. All subsequent antibodies derived this way trace back to a unique parent cell. Monoclonal antibodies have monovalent affinity. As it is monoclonal, so it has the monovalent affinity. It binds only to the same epitopes. What is epitope? Epitope is a part of an antigen that is recognized by the antibody. The next question is, who first invented polymerase chain reaction? That is the PCR reaction. Option 1 is Paul Ehrlich, option 2 Jill Nielsen, option 2 Kerry Mullish, and option 4 is Porter. And the correct answer is Kerry Mullish. PCR is a technique that rapidly produce or amplify millions to copies of specific DNA segment, which can then be studied in greater details. It involves using a short synthetic DNA fragments that is known as primer to select a segment of the genome to be amplified. Then multiple rounds of DNA synthesis to amplify the segment. Thank you. Please subscribe to this channel.